research in life sciences, so that startups with an interesting idea can come and straight away exploring their idea and see develop it to such an extent so that they can go to the market or ask for investments to take it to the next level. So we are primarily encourage startups that are at the idea stage to proof of concept stage. So that's what I've been doing for the last 5 years. And I think we are doing okay. But one of the other things that we wanted to make an impact is, one of the initiatives we have taken is the CPHMS. I know it is a mouthful, but it is called the Center for Predictive Human Model Systems. This center formed while we were discussing with another organization called Humane Society International, which was interested in developing or promoting, more humane ways to do research. One of the humane ways to do research is, can we look at, do we really need to use animals to do certain amount of work, or is there any alternate to usage of animals in research, whether it is drug development or mostly in drug development, or to understand diseases, so on and so forth. So the idea was, the reason for initiating this center was based on two reasons. One of them is, by then, we know that the current models to evaluate drugs that can eventually be used in humans, the models are not really useful. Their utility is compromised for various reasons. Like most of the time when I talk about models, we're talking about mostly mice and rats, because as you go up the scale, like rabbits, dogs, and monkeys, they become very expensive. But one of the things that we can really screen a bunch of drugs for their efficacy in mice and rats. And we've been doing it for the last several decades, maybe 7-8 decades. But the kind of knowledge or understanding that today we have is these models are not very effective in recommending the right molecule to go to the next level. Which means that then why are we doing it? When there is certain amount of similarities between the mammals, but is that similarity sufficient to really evaluate a drug's efficacy and use it straight away in humans? That was never a comforting thought. So people are always looking for alternates. So one of the things that we initiated was, when we looked around the activities in this particular direction across the globe, people are using a lot of in vitro model systems to evaluate the drugs. For example, I kind of come back to the context of drug development in my next few minutes, just because it's convenient to talk about. For drug screening, there are alternate models, but the awareness in India of these models is not very high. There is occasionally here and there some people are using them, but one of the things that we thought, we should promote these alternates to in vitro models to see and educate people, and also impress upon the policymakers that there is a good opportunity to reduce the usage of animals and replace the animals, so that we would be able to get the same information probably. So this is something. At the same time, elsewhere in the world, people are started developing, are also a bit wanted a more appropriate models for their own work, research work. For example, if I want to investigate something on a liver, probably till earlier times, people were isolating the liver, primary hepatocytes or some liver-derived cell lines and use them for their investigations. But that, they are not happy because many of the pharmacological effects of a drug, simply doesn't depend on a single homogeneous cell line. Liver is a very complex organ. So there are several cells that make a liver. So when you want to look at the pharmacological properties of a drug on a liver, you want to recreate the liver. So one of the things that people started looking at is, can I recreate a liver, something similar to liver in vitro? So that's where these terms like organoids came into play. Now, organoids are a kind of next level complexity created by us so that in a multi-layer three-dimensional system, one can investigate the pharmacodynamics, how an organ interacts with the drug. So this is something that really excited people. And it definitely, because in certain, if somebody is smart, they can create a vesicularity in the organ also. So it almost like a kind of a miniature liver in a test tube. These things were very successful and people were very happy with the kind of results they are getting. And then the next thing that they wanted to go for is, can I reproduce not only one organ, but many organs together in a single system? So that's where people came up with these microphysiological systems, or organs on chip, or tissue on chip, or body on chip, whatever you may want to call them. There are several of these things they came up with. And there is a huge interest. See, these days, science has become very interdisciplinary. Now to do a organ and chip, you would like to talk to, people from microfluidics, or what we call electromechanical devices, how to print things in a 3D pattern, so that you can do entire thing on a something like a matchbox kind of a size. You can create a large number of organs, at least 4 or 5 organs in a chip, and create a kind of fluidic systems, almost something that mimics the blood, and then investigate and see whether there is an opportunity to come up with a kind of understanding that is very similar to what happens in a complete organism. So these microphysiological systems excited everybody because it's a kind of a interface between chemical engineers, designers, biologists, medical expert, pharmacologists. So this is a very exciting area that came up. And this is where our the context of today's presentation is. That is about microphysiological systems, their applications, and so on and so forth. And our purpose, as I said, is to increase the awareness, educate people, try to find some funding, and bring in a lot of policymakers or the regulatory people to appreciate the new developments and write a lot of white papers and put them together in the same room so that there is a better understanding and implementation of these new knowledge in today's world. Because if you look at Europe or US, people are already looking at it very seriously and planning to replace the usage of animals in drug development, for example. So, am I audible or am I breaking? Okay, anyway. So I'm coming to an end of my whatever I wanted to say. This particular conference or the lecture series. The lecture series, you should remember the word lecture series because we planned it that way, where we bring in the best in this particular game from Europe and India, and put them together along with the young people like you, so that there is a kind of one-to-one interaction between the experienced researchers and the young people who are interested in microphysiological systems, so that this particular area will get a kickstart. And we are very happy that EMBO recognized our efforts at CPHMS, in promoting microphysiological systems, and invited us to conduct their one of their lecture series here. So, for those who are, heard about microphysiological systems vaguely, or want to know more about it, want to get into it, please do register and come to our conference. It'll be a great opportunity to rub shoulders with one of the best that come from Europe and also from India, because this is something that's going to stay for a while, for at least for a decade, because it's a developing area, and there would be a lot of interest. And I can tell you, it's interest not only from biologists, but as I said, people, pharmacology people, and fabrication, microfluidics. It's a very interesting and exciting interface, almost the kind of excitement people had when nanotechnology came into play. So it is something similar, and I'm sure you would find the kind of topics interesting, and the people that are coming are very experienced. And rest of my team, like Surat, Kadambari, and Anushka, will be speaking more about the details and operational details of this conference. And I wish you will give yourself a chance to look at the details and see if it falls in your path. We will really encourage you to apply for it. Thank you very much. And if you have any questions, we are around. I think once my colleagues finish their presentation, we'll come back for Q&A. Have a nice day.
Thank you so much, sir, about this insight over CPHMS and microphysiological systems. So now I'll just go through the definitions of microphysiological systems. Sir has covered most of it, but I will start by saying that microphysiological systems are basically an interconnected human cell-based platforms, and these are designed to recapitulate the physiology and function of human body, which is in vitro and at a micro level. Now, these are often referred to as either organs on chips or organoids. And organoids are basically the miniaturized and simplified 3D versions of organs which are produced in vitro. Now, certain characteristics about these organoids are that they are self-organized, and thus we can study processes like organogenesis on these organoids, which can be done in vitro. Now, these are 3D organoids which give us a better view about an organ as a whole. And moving on to organ on chip, it is a clear, flexible polymer micro device, and it contains hollow microfluidic channels which are lined with cells. And certain organs on chips also provide mechanobiological functions, and they are being developed to provide functions like vascularization and mechanobiological functions. Like, for example, if we have a lung organ on a chip, we can give channels where there would be contraction and relaxation, how the lung will show while respiration is happening. So this is about microphysiological systems. Now, this course will talk about microphysiological systems as a whole, and it will be a 5-day course where we have speakers from India as well as international speakers coming in Hyderabad from 31st October to 4th November. And the course will have various sessions. Just 1 minute. Yeah. So talking a little bit about the course, we will have sessions which will be about how this modeling of human development. Now we have different sessions where people will talk about modeling a brain as an MPS system, modeling tumor, modeling cancer, so that we can talk about how to overcome those challenges using microphysiological systems. Then we will also be having speakers who will talk about the current challenges while developing microphysiological systems. For example, how to develop vasculature in these systems, how immunization can be included in these systems, and also topics like scaffolding, microfabrication, which include the designing of these organs on chips and organoids, will also be discussed. Then, as Sir said, microphysiological models are used in drug discovery. Even those will be discussed in one certain session. And another session, we will talk about mechanobiology, which I mentioned before. So, we have speakers like Dr. Abhijit Muzamdar, we have Donald Ingber as one of our virtual speakers, and these are scientists who have worked great in microphysiological systems. So I'm going to call my another colleague to talk about how you can register for this course and how you can talk about your own research in this course. So let me invite Anushka to talk about this.
Hi, everyone. Good morning. So I'm going to be speaking to you about what the conference, what you're going to get from the conference, how to register, and some of the exciting things that we have lined up for you. So one of the first things is the course objectives, which I just want to go over. So the objective of this lecture course is really to help all the participants understand what microphysiological systems are about, understanding their design and development, so that anyone who is currently working in the field or anyone who wishes to work in the field can gain insight. Then we have one of the second objectives is to understand what are the current challenges in this field, because we have a lot of researchers who are making great waves in microphysiological systems and in the 3D cell culture or organoid research. So they understand what are the most important challenges, and they'll also be speaking about how to address those challenges so that we can have expert opinions on how it would be easier to bring this kind of research into the Indian landscape. And I think it would be very useful for anyone who is planning to pursue work in this field. Then we'll also be helping you understand translation of these systems from the lab to industry. And finally, there'll be a lot of mentoring sessions, panel discussions, and some practical demonstrations, which is a great opportunity for PhD students not only to interact and network, but also actually take a look into what these organoid cell cultures and organoid chip systems are. Now, a little bit about the application process. So I'll be adding the link to our website in the chat box, so you can check out our website as well. We have all of the information about the lecture course. It has a detailed kind of itinerary of the day-to-day sessions, the speakers. You can check out the profiles of the speakers as well. And the things that you will need to apply is a CV, a motivation letter, and a reference letter from your PI or mentor. Now, here we've mentioned that the eligibility is in 3 categories: students, faculty, and industry. And a lot of people have asked us that if there are a lot of master students, a lot of junior research fellows and research associates who were interested. So we would like to say that we do encourage you to apply if your work is relevant to the field, or if you are interested in learning about microphysiological systems. Even if you don't have a master's degree, even if you don't have a PhD, we recommend you to apply, because we will be evaluating all of your applications based on how strong your letter of motivation is, and based on your background and whether you've done some work in the field before, or whether you're planning to do some work. So basically, your motivation letter is what is going to kind of help you get selected. So we do recommend anyone and who's interested in this field to register. And just so we can make coming to the conference a little bit easier and a little bit more accessible, we are also offering a limited number of waivers and grants. So we have travel and accommodation grants. We are also offering registration waivers. So if you don't fall into one of these categories completely, let's say you work at a non-profit or you work at a startup and you would still like to attend our conference, we do encourage you to apply in whatever section is closest to what your qualification matches or whatever your designation matches. And we urge you to apply for the registration waivers and for the travel and accommodation grants. There is also childcare support for anyone who wants to attend the conference and cannot leave their children behind or want to bring them with. So we do offer these kind of support systems. And we will, again, you have to provide a reason as to why you're applying for these waivers and why you're applying for the grants, and a committee will be evaluating all of your applications. Now, one more thing that I would like to talk about is the poster presentation and postdoc talks. So we are also accepting abstracts for students, for PhDs, for postdocs to present posters at the lecture course. And we will also have a few slots for postdoctoral students to present talks about any work that they're doing which is relevant to the theme of the lecture course. So again, that is also open. I think I've not mentioned this yet, but our registrations are open until the 10th of August. So even abstracts are, you can send in your abstracts until August 10th. After that, we will not be accepting any more applications. So you still have a little bit of time, but I encourage everyone to check out our website, check out the different speaker profiles. You can see the itinerary. You can also visit the CPHMS page, which is found on the Atal Incubation page, and you can check out our blogs. You can check out our previous newsletters to get a little bit more of an idea about what is the kind of work that we are doing, what is the importance of human relevant research in microphysiological systems. And we are also hosting a competition. So we are hosting a storytelling or a story writing competition and a sort of photography / image competition where we are encouraging people to present to us their take on microphysiological systems in the form of a story or in form of any cool images you might have taken while working with organoids or 3D cell cultures or organ on chips. So there are some exciting prizes for the winners, and you'll also be featured in our upcoming newsletter. So we do recommend you to apply for all of these things. We're trying to make it as fun as possible and as interesting and engaging as possible. So thank you everyone for your time. And now I think we'll open the floor for some questions. So if anyone has any questions, please do put them in the chat box. I'm going to just address some of the questions which have already been sent. You guys can keep sending them in. So the first, I think I would ask Dr. Madhu to maybe come back on. So you there?
Yeah.
Yeah. So the first question is, what is AOP?
What is the
AOP, adverse outcome pathways?
Yeah, adverse outcome pathways is primarily a document that kind of puts together in a very structured format all the details that people have observed with a drug, so that as you know, adverse outcomes are the most crucial things that play a big role in evaluation of a drug. So documentation, because before a thing becomes interesting for drug as a drug, one needs to build up a lot of people must have generated a lot of data on it. So one of the I think European Medical Agency has come up, and several other organizations have come up and said, let's put up a standard format wherein all the information regarding a drug is at one place. These are the things that would help to systematize the knowledge on a particular drug and help us to understand it better, and then look at well-curated data. That would be very useful. So these documents, we encourage people to come together so that to put it together so that it will be a kind of a reference document for a particular drug. Right? I mean, all the scattered information curated and put together And that would be a kind of a reference material for the drug. Like you may have reference chemical libraries, chemical documents for a drug. Like that, this will be more on the pharmacological aspects of it. Right?
Thanks, sir.
Thank you, sir.
I hope that answered your question, Ami Mehta. Next question, I think I've addressed this already. Master students, yes, we do encourage you to send in your applications and register. So Asha has asked, is it an offline only course? Yes, this is an in-person event which will be happening in Hyderabad, October 31st to November 4th. We are not having a hybrid or an online session. Then the deadline for application, yes, deadline for applications and abstract submission is August 10th. For the competition, I'll be sharing a link again. I'll be sharing a link below. You guys can check it out there. The deadline for the competition is July 31st. It is not August 10th because we will also be coming out with our newsletter soon. So we would like to have your entries before we do that. Okay, someone has, yeah, so here's, you guys can take a look. This is about the competition, and I've also put the registration link in the chat box for the competition. Next we have accommodation for participants. So there are currently 2 registration fees. There is one with accommodation, without accommodation. If you would like us to arrange your accommodation, you can register for that, and you can also apply for an accommodation waiver. Otherwise, you can, if you would like to not avail our accommodation, you can always just register without that and make your own arrangements. Then Shri Harsha has asked, how do we send the reference letter from the PI? Should it be uploaded while registration? Yes, so once you register on the website, there are places for you to upload all of your documents. So you can do that on the website itself. Again, the registration is also on the website, the EMBO microphysiology website, the link that I shared previously. So please scroll to the bottom. You can go to the, maybe I can just show you guys once where everything is. So this is our website. If you click here and you go to registration, okay, maybe I'll show you guys the entire website just so everyone can have a look. We have some information about the course. These are all of the important dates that you need to keep in mind. So the registration and abstract deadlines are on August 10th. Then we will notify selected participants by the 25th of August, and you can finish all your payments. You have to finish all your payments by the 15th of September.
Sorry.
Could you guys please mute your mics? Thank you. So you can also find our social media handles, and you can download a copy of this poster if you would like to share it in and around your institutes. And then if you want to kind of tweet about us, go ahead and use this hashtag that we have. We have a list of all the speakers. You can click on their names. It'll redirect you to their profiles, and you can take a look at what all of our speakers are working on. Then we have, like I said, we have an itinerary here. So you can, there are 5 days of the program. You can see what all of the sessions will be, what day, what timing. You can plan your itineraries accordingly if you do plan to come. Now, again, we have information about the registrations. So here are all the registration fees and the information. And this is what you're going to click to apply. So you click online application and the apply now thing will come up. And that will take you to another website where you have to register and submit your application. And there's a whole bunch of information here. So everything is kind of laid out here, your poster specifications, information about the waivers and the grants. And if you would like to contact us about any questions, there's our email over here. So please do check out the website. All the information is on there. Some more details about the storytelling competition. Kadambari, would you like to take this one?
Yeah, sure. So we are asking for articles, any popular stories or anything you have encountered about microphysiological systems before. You can submit a short article about it on the link sent in the chat box. And we need this before 1st of August. So 31st August is the last day to submit the article. And we're also inviting pictures like organoid pictures or any kind of art you might have on microphysiological systems to be submitted in this competition. And winners will be featured in our newsletter and on our website. Organoid pictures would also be featured in the India EMBO lecture course abstract book. And I encourage all of you to apply. We also have exciting prizes, like we are giving Amazon vouchers to the winners. So there is a link in the chat box, and if you take a look at that, you will find further details. Thank you.
All right, next question. I think Dr. Madhu, this one is for you again. So Khemraj Thakur has asked, our lab is planning to shift to in vivo bracket mice or rat/animal experiments of microbes to organoids. Can we do it in microphysiological or organoid systems?
I'm just looking at it.
Yeah.
Which one is this?
Khemraj Thakur?
Okay, yeah. Shift in vivo. I just need the question to be a bit more expanded in the sense experiments of microbes.
Khemraj, maybe you could elaborate a little bit about your question and send it again in the chat box.
I'll just come back to it.
Yeah, we'll take some of the other questions till then. Divpreet Kaur, does registration include accommodation? I think I just went over that. We have two registration kind of options, with and without accommodation. So you can take a look at which one you would like to choose. Mamta Mishra has asked if anyone has started blood-brain barrier research in microfluidics in India.
Yeah. Not to my knowledge yet, because there are a couple of groups that work on, what kind of technology need to be used to overcome the blood-brain barrier for a larger molecules. For small molecules, it's not a problem. But to my knowledge, not so far. Nobody has created that vascular system yet. But it is very similar to other evolved, barrier systems like skin or lung epithelial and things like that, but with a different tight junction properties. Because blood-brain barrier has a tight junction property that are very different from the other tight junctions elsewhere in the body. I'm not sure, but we can come back to you if you send an email if I come across anybody who is doing it now. Okay.
Okay, let me just take a look at the next question. So Ami Mehta again has sent a question. What if you have submitted PhD thesis and not started with postdoc yet? Are you still eligible? If so, under what category? So PhD students and postdoc students come under the same category. You will be eligible. You can apply, and I believe that if you are a student, you have to send a reference letter. So you could ask for a letter from your previous mentor. I'm sure that'll be okay. So yes, you are still eligible. You can apply. Is the abstract submission mandatory for all the participants? No, Sushmita, it is not mandatory. Anyone who is interested in presenting their work. So I do want to say that if you are submitting a poster, we are looking for people whose work is very specific to the theme of the conference, or even if you have any ideas on how using microphysiological systems or organoid systems could address a research question that you have, and you've done a little bit of insight, you've done some background work and you'd like to present that, I think that will also be okay. But we are not accepting abstracts or poster submissions from something that is completely unrelated to the theme of the conference. So I would like to stress on that. It should be related to our conference theme. But no, abstract submission is not mandatory. It is only for those people who are interested. Okay, Khemraj Thakur has elaborated. Specifically, we want to perform biofilm formation of Candida auris or C. glabrata in organoids instead of using animal models.
Yeah, I mean, definitely it looks, but even in an animal model, the formation of biofilm must have a specific organ in their plan. Like, if they want to look at biofilm formation in the GI tract, for example, the target organ is very important, obviously, for their work. So if they have decided on the target organ, definitely there are organoids for most of these organs today. So in fact, it will be a kind of a better approximation, or it's something that are much more easier to do than an animal model, than in organoids, because these are all controlled systems. Unlike an animal model where there is many other factors may mitigate your confound your results. But here in organoids, it's much simpler. I think it's not a bad idea to start not only for control conditions, but also for the reasons of cost. If the moment you say animal models, you have to take permissions. Somebody else will do your experiment. You'll have to go and collect the tissue. Instead of that, organoid models gives you control that you can that's in your hands. Probably it's not a bad idea to start doing in organoids. Right?
Okay.
If you still have some, we can come back to
it.
Yeah.
I think if you have any more questions, I think if sir is all right with it, you can contact sir and maybe ask any more questions. If you want to elaborate your question a little bit more here, feel free to send in your question again, Khemraj. There's one more question, sir, from Karuna Nagula. We have an animal cell culture lab already established. Can we convert it to microfluidics, organoids, spheroids culturing? Are there any guidelines for that?
Yes. But anyway, for organoid, spheroid kind of a thing, somebody must be having some expertise on it in your institute. That's first thing, because it's just not a linear, the next step in animal cell culture is organoids. One needs a bit more understanding of the biology of organoids. So that's one thing that you should have. When it comes to microfluidics, probably you can buy off some chips that are suitable for your purpose, because there are companies that are selling a lot of microfluidics. So chips for various purposes, depending on the complexity, you may choose one. And obviously you need to have some kind of a fluid management system, like, the controlling the flow of liquids through the various chambers of a chip. So you just cannot convert it. You need people from microfluidics, some other departments, and somebody who has already done some organoid work, and then you would be able to get to it. Otherwise, simple thing is collaborate with somebody who's already doing, so you can transplant their know-how to your animal cell culture lab. The expertise is required are different. So it is usually in an animal cell culture lab, the experts just simply cannot convert it into a organoid microfluidics lab or a microphysiological systems lab, just like that. It needs some effort. Right?
Right.
Thanks, sir.
Thank you, sir.
I think Dr. Radha has also sent in a comment. So Dr. Radha is also one of the speakers at our conference, and she's also one of our co-organizers. So she's just said that you need to define the purpose of making the organoids. So if Dr. Radha, would you like to elaborate if you want to just pitch in? Just wait a few seconds. Let's see if she's.
Yeah, now I can. Yes. Hello. Yeah. Since you already have a cell culture facility, you have to be clear about why you want to make the organoids. Is it for a drug discovery purpose, or just to make a platform or test out your ability to make these organoids. So once you have that definition clear, then I think you can work towards it. Or is it just for a demo purpose? Unless you know why you want to do it, and also which kind. Some organoids are very difficult to make. Some tissues are relatively easier to generate organoids. Is that clear?
Yes, all right. Thank you, Dr. Radha.
I hope that addressed your questions, Karuna.
And would you like me to mention about, somebody had a question about the blood-brain barrier.
Yeah, ma'am, sure, go ahead.
Okay, so right now the answer is no in India, but there's a lot of effort going on to co-culture endothelial cells along with differentiated iPSCs that have taken the neuronal lineage. So you will have these mini brains formed, which also have endothelial cells. But that's at the simplest form of looking at a blood-brain barrier. We are not really there yet as to understanding the blood-brain barrier or utilizing the system to examine blood-brain barrier in the organoids. Okay, but then there is an effort of looking at what can cross the endothelial cell films, or monolayers of endothelial cells, which actually mimics. So you can have a monolayer of endothelial cells, and then you can have your brain organoid sitting just beneath or above, and see if you can pass molecules through it. We are really at an infancy in this domain. Okay.
Thank you, ma'am. Okay, so we have one more suggestion from Sri Harsha. Many of the IITs and ISC have independent organ-on-chip efforts, which are mostly unknown to other colleges. An effort to bring them all together on a single platform might be helpful to the community. We have encountered many trivial problems, which might have been solved by other groups, so that everyone doesn't invent the wheel again. A great suggestion. I would like to mention that this is one of this is something that we are working on as well. This is one of the main tenets of our center as well. So maybe I'll let Kadambari speak a little bit more about what we are doing to create a more integrated and cohesive network of Indian researchers who are working in the field of human relevant research and microphysiological systems. So over to you, Kadambari.
Just before that, I think we have one document made where we looked at all kinds of activities, projects done. Probably, Kadambari, are you going to say about it?
Yeah, I was going to talk about the open access database.
Yeah, just go ahead.
Yeah, so we have contacted researchers, and if they were working on organ-on-chip or any microphysiological systems, we have created a database about them, and we are planning to create this an open access database. We will be publishing it on our website soon. So currently, we're also going to reach out to more researchers to get their details, and it will be an interactive database live on our website where not only the researchers will mention what they're currently working at, but also what they plan to do and where they might need expertise. So this would be a platform to invite collaborations between researchers. So I just suggest that all of you can stay tuned for CPHMS to bring this to you. Thank you.
And, it's also an open call for everyone who is working in this field. We really want you to come and join us, be a part of our database, so that we can create a really strong network of Indian researchers who are working in this field. And I think, like Sri Harsha really made a great suggestion. It would be easier to solve problems within the community if there is a very strong community. So there's also one more thing I'd like to share here. I've shared a link in the chat box. These are some of the white papers which we have published, which focus on, sorry, I think my voice got frozen for a second. So we've published a few papers about developing these systems in India. So developing adverse outcome pathways and microphysiological systems. and even systems and computational biology. And we, a lot of these things are also mentioned in the newsletter. So I really urge everyone to please check out our website. We have a lot of resources available for anyone who's interested in learning about human relevant research and microphysiological systems and so on. So there's a comment from Mamta Mishra for you, Dr. Radha. Yes, the successful co-culture of neuron glia is done in many labs, including me. Endothelial cells together will be an addition towards the blood-brain barrier. Thanks. So thank you everyone for your questions. If there are any more, please do send them in quickly. I think there are only 5 minutes left. Other than that, yeah, I think I'd just like to say thank you to everyone for joining us today. We do have this session available on YouTube, so I just want to share that link with you guys as well. You can also check out our social media pages, because we will be sharing this information across our social media pages, but the link is here as well if you would like to share it with people people who are working in the field who couldn't make it to the webinar today. So they can get a little bit of insight into what the India EMBO lecture course is going to be about, why you guys should come and why it's really important for everyone to come and network and create this great integrated community. So I think that is all for today. Again, please do check out our website. Please do check out the India EMBO conference website so that you can get all of the details. If you have any questions or queries, you can reach out to us at the following email IDs. I'm adding those in the website as well. In the chat box as well. So for those people who don't know, my name is Anushka Banerjee. I am a research associate at CPHMS. So you can always contact myself or Kadambari if you have any questions. I think that's all. So, we hope to see you there. Go ahead and, put on your creative caps, apply to our contest. There are some great prizes. It's going to be a great 5 days in Hyderabad. So we're looking forward to having you here with us. Thanks, everyone.
Thank you, everyone.
And thank you very much, Madhu sir, for being with us this morning. Sorry, sir, you muted. Yeah. If you have any closing words.
No, I mean, I'm fine. I'm sure we kind of tickled them a bit on microphysiological systems. Hope they'll come back.
Okay. Thank you, everyone.
Yep.